Hello friends, in this lecture we will learn and discuss the various types of culture media. On the basis of utility, media are classified as minimal media, selective media, differential media, selective differential media, enrichment media and enriched media. Let us discuss each of these one by one. The first is minimal medium. Minimal medium is a medium which supplies only the minimal nutritional requirements of a particular microorganism. All minimal media are chemically defined but not vice versa. Minimal media are useful in the study of nutritional requirements of the microorganism. Since minimal media are formulated keeping the nutritional requirement of one study microorganism in mind, different minimal media are required for different types of microbes. Here is the composition of a minimal medium for Bacillus megaterium. Let us move on to the next type of media, that is selective media. On a general purpose nutrient agar media, we get the organisms which are predominant in that sample, but not the organisms which are maybe desired by us. This is because the organisms desired by us are simply outnumbered. This can be rectified by designing a media which selects and allows the growth of only the desired microorganism and inhibits the growth of all other microbes. Such a medium is called a selective medium. A selective medium supports the growth of desired organisms while inhibiting the growth of many or most of the unwanted ones, either by purposely adding one or more selective agents which poison certain types of organisms, or by including or deleting certain nutrients such that the desired microorganisms are able to grow better. Let us discuss a few examples of selective media. The first is McConkey agar. This is an example of a medium where selective agents are added which directly suppress the growth of undesired organisms as much as possible. The particular selective agents chosen for this medium are bile salts and crystal violet, which inhibit gram-positive bacteria, thus allowing the near exclusive growth of gram-negative bacteria. The next example of selective media is mannitol salt agar. It is a selective medium for members of the genus Staphylococcus. The medium contains high concentration of salt and Staphylococcus can tolerate high saline levels. Organisms from other genera may grow, but they typically grow very weakly. In the picture, we can see that Staphylococcus aureus grows on this medium, but Serratia marcescens, which cannot tolerate high salt concentrations, does not grow. The next example of selective media is phenyl ethyl alcohol agar. It is a selective media used to cultivate gram-positive bacteria. The selective ingredient phenyl ethyl alcohol inhibits the growth of gram-negative bacteria by interfering with DNA synthesis. In the picture, we can see that Staphylococcus aureus, which is a gram-positive bacteria, grows on this medium, but Serratia marcescens, which is a gram-negative organism, does not grow. Moving on to the next type of media, that is differential media. Differential media do not inhibit the growth of any microbe, but as the name suggests, it helps to differentiate between types of organisms growing on the same agar plate. For example, in this plate, you can see green colonies as well as pink colonies on the same plate. This may be due to dyes and other components added to the medium upon which different organisms act in different manner such that the end products are different, as you can see in this particular agar plate. This can be detected by the variation in color. In this agar plate, we can see different colored colonies all on the same plate. Let us discuss a few examples of differential media. The first example is lactose fermentation broth. The medium is differential because of the presence of lactose as a carbon source and bromocresol purple an indicator dye. Organisms capable of carrying out fermentation of the lactose to acid change the pH of the medium, which causes the bromocresol purple in the medium to turn from purple to yellow. Those bacteria that are unable to ferment lactose will still grow if another carbon and energy source is provided that they can use, but no color change will be observed in the broth. Thus, we can differentiate between lactose fermenters and non-lactose fermenters using the same medium. The next example of differential media is starch agar. Starch agar is basically an all-purpose medium like that of nutrient agar or heart infusion agar, to which 0.4% soluble starch is added. This media differentiates between organisms which can utilize starch by producing amylase. After incubation, the medium is flooded with grams iodine. The iodine reacts with starch to form a dark blue colored complex. Any clear area around the growth of the culture after the addition of the iodine indicates the breakdown of starch by the organism due to the production of amylase, as can be seen here. Those which do not show any zone of clearance are amylase non-producers. Moving on to the next type of media, that is selective differential media, which not only selects a particular class of bacteria but also differentiates within them. The first example of this type of media is eosin methylene blue agar. This is commonly used selective differential plating medium for the detection and isolation of gram-negative bacteria. It is especially used in the isolation of coliforms from water and food samples. Eosin and methylene blue are the selective agents which inhibit the growth of gram-positive bacteria. The lactose and sucrose contained in this medium allow lactose and sucrose negative Salmonella and Shigella to be distinguished from lactose positive coliform organisms and lactose negative sucrose positive accompanying flora like that of Proteus vulgaris, Citrobacter cosseri and Aeromenas hydroflora. Lactose fermenters like the coliforms form dark colonies due to the acid produced by fermentation. Classical types of colonies are seen on a typical EMB agar plate. They are dark, almost black colonies that are of coli type. By reflected light, these colonies show a green metallic sheen, which is due to the precipitation of methylene blue in the medium from the very high amount of acid produced from fermentation. Those which form this type of colony are mostly strains of E. coli and some strains of Citrobacter. The second type of colonies are dark-centered colonies surrounded by a light mucoid rim, because of which it appears a fish eye type of colony. Most strains of Klebsiella and Enterobacter show this type of colony growth. The third type of colonies are colorless colonies. Non-lactose fermenting colonies produce no acid from fermentation, so the lighter colored alkaline reaction is seen. These colonies appear colorless like that of Pseudomonas. The next type of selective differential media is xylose lysine deoxycholate agar, commonly called as the XLD agar. XLD agar is another selective differential agar used to differentiate Salmonella and Shigella from coliforms. This medium contains 3 fermentable sugars, xylose in less amount and lactose and sucrose in larger amounts. Apart from this, it also includes an amino acid, lysine, which when decarboxylated causes a significant alkaline reaction. The pH of the medium initially is 7.4 and the indicator used is phenol red, and hence the medium is pink red color initially. Salmonella can ferment xylose and produce acid and hence become yellow. But once xylose is depleted, it decarboxylates lysine to produce an alkaline reaction which over-neutralizes any acid produced by xylose fermentation. Salmonella also metabolizes thiosulfate to produce hydrogen sulfide, which causes the red colonies to become black-centered, as can be seen in the picture here. Shigella cannot ferment the sugars but can decarboxylate lysine. However, they cannot metabolize thiosulfate. Hence, black-centered red colonies of Salmonella can easily be differentiated from the red colonies of Shigella. Coliforms can ferment lactose, which is in large amounts, and hence the net reaction is acidic and the colonies are yellow colored. Hence, they can be easily differentiated from Salmonella and Shigella colonies. XLD also contains sodium deoxycholate as the selective agent, which does not allow gram-positive bacteria to grow. The next example of selective differential media is McConkey agar. McConkey agar is a very common selective differential agar used to identify lactose fermenting gram-negative enteric pathogens. The bile salts and crystal violet in the media selectively inhibit the growth of gram-positive bacteria, allowing only gram-negative bacteria to grow. This media is used particularly to distinguish between lactose fermenting and lactose non-fermenting bacteria. Lactose fermenting bacteria like E. coli and Klebsiella ferment lactose and produce acid, which lowers the pH and the colonies appear red. Non-lactose fermenting bacteria like Salmonella and Shigella will not ferment lactose but will use peptone and produce ammonia. This increases the pH of the media and the colonies appear colorless. The next type of media on the basis of its intended use is enrichment media. Enrichment media was first introduced by Martinez Beijerinck. When a particular bacteria from the soil is to be isolated, it is not easy to do so by streak plate or pore plate on a general purpose medium like nutrient agar. Bacteriologists make use of a medium called enrichment medium. The medium is designed according to the nutritional requirements of the desired organism so that its number increases to observable levels. The medium will omit a nutrient needed by most undesired bacteria or include something that only the desired class of microbes can use. Media of this type are typically formulated to mimic the environment of the desired organism or will take advantage of a known metabolic property of it. Enrichment media differs from selective media in that no selective agent is added to suppress the growth of undesired organisms. The medium, therefore, simply favors the growth of desired organisms. Suppose we need to isolate a phenol-degrading bacteria from soil. A media in which phenol is the only carbon source is made and inoculated with the soil sample. Only phenol-degrading bacteria can grow utilizing phenol and all other bacteria are inhibited. Similarly, if nitrogen-fixing bacteria are to be isolated from soil, a media with all the other components except a nitrogen source is made. When inoculated with the soil sample, only nitrogen-fixing bacteria can grow, which can assimilate the atmospheric nitrogen. A selective enrichment medium for the growth of extreme halophile, like that of Halococcus, contains nearly 25% salt, which is required by the extreme halophile and which inhibits the growth of all other organisms. The picture here shows an extreme halophile isolated from Dead Sea. The number of salmonella in a fecal sample may not be sufficiently high to give a positive test. The sample can be cultured in a selenite F broth containing selenium as a trace element which supports the growth of salmonella. Moving on to the next type of media, enrichment media. Enriched media. Certain fastidious organisms like Haemophilus influenzae, Actinobacillus, Botydella pertussis, Brucella melitensis, Campylobacter fetus and Helicobacter pylori do not grow on ordinary nutrient medium. They require growth promoting ingredients such as blood, glucose, serum or egg. Addition of these extra nutrients to basal medium makes it an enriched media. Let us study a few examples of enriched media. The first example of enriched media is blood agar. Blood agar is an enriched medium in which nutritionally rich whole blood supplements the basic nutrients. Used for isolation and cultivation of many types of fastidious bacteria. It is also used to differentiate bacteria based on their hemolytic characteristics, especially within the genera Streptococcus, Enterococcus and Aerococcus. Several species of gram-positive cocci produce exotoxins called hemolysins able to destroy red blood cells and hemoglobin. Blood agar, which is a mixture of nutrient agar and sheep blood, allows differentiation of bacteria based on their ability to hemolyze RBCs. The three major types of hemolysis are beta hemolysis, alpha hemolysis and gamma hemolysis. Beta hemolysis is the complete destruction of RBCs and hemoglobin and results in a clearing of the medium around the colonies, as can be seen in this particular picture. Alpha hemolysis is the partial destruction of RBCs and produces a greenish discoloration of the agar around the colonies, as you can see in this picture. Gamma hemolysis is actually non-hemolysis and appears as simple growth with no change in the medium. The next example of enriched media is chocolate agar. Chocolate agar is a variant of the blood agar plate. It is enriched with heat-treated blood, which turns brown and gives the medium the chocolate color, as you can see in this picture, for which it is named. It contains red blood cells which have been lysed by heating very slowly to 56 degrees. Chocolate agar is used for growing fastidious respiratory bacteria such as Haemophilus influenzae. These bacteria need growth factors like NAD and hematin, which are inside erythrocytes. Thus, a prerequisite to growth is the lysis of the red blood cells. The next example of enriched medium is Loeffler's serum medium. The medium is enriched by the addition of sheep or horse serum to glucose in the ratio 3:1. It gives rise to an opaque whitish solid medium. Serum is sterilized by filtration. It is used for cultivation of Corynebacterium diphtheria. Yet another example of enriched medium is Lowenstein-Jensen's medium. This medium is enriched by the addition of eggs. 1000 ml beaten eggs are added to 600 ml of Lowenstein-Jensen's medium base, which mainly is made up of monopotassium phosphate and magnesium sulfate and L-aspargine. The medium contains malachite green as selective agent and hence is green in color. It is used for isolation and cultivation of Mycobacterium tuberculosis, which forms non-pigmented, rough, dry colonies on the Lowenstein-Jensen's medium.